Welcome to Tactic Academy, where we'll be looking at Gram-Stein procedure or Gram-Stein experiment. So it's an experiment usually conducted by scientists or doctors to say. All right. So a Gram-Stein procedure or a Gram-Stein experiment will be conducted. So what is this Gram-Stein experiment? Let's get started. All right. So a Gram-Stein procedure or a Gram-Stein experiment is a test that checks for a bacteria at the site of infected area, all right, or infected infection. So let's say you have a wound here, all right. Here is a person, or just here is a hand, all right. So so this is a person's hand, all right. Let's say there is a survey, or we can say a big wound here, or a sore to say, all right. So this sore it is growing, all right. So we need to know the type of bacteria as scientists so that we can be able to know the type of treatment that we should use, all right. So now this is where we say a Gram-Stein procedure is a test that checks for a bacteria at the site of infected suspected infection. All right. So what is the purpose of this Gram-Stein procedure? So we'll say, I'll define it to say is a test that is a test that checks for a bacteria, all right. So it will be there to check for a bacteria at the site of what? At the site of suspected infection, all right. So a Gram-Stein procedure or a Gram-Stein experiment, so at the site of who are suspected infection, all right. Okay. So this is a test that doctors will conduct in order to know the type of bacteria that is there. So the purpose of so the importance, all right. What is the purpose of us performing this Gram-Stein procedure? Sometimes not just for a treatment, it's also for a study, so that for further treatment can what's this knowledge can be added for further treatment. Okay, so the purpose of Gram-Stein procedure is to discover, let's say, a new species or a new virus, because we'll be able to know the type of virus that we have today. It has possessed this character. So the first point that we are going to say, we'll say the importance of this guy is one, to discover a new species, all right. Discover the new species, or just a new species, discover of new species, all right. Or just in research, all right. So we'll be using this in research, or just for discovery of new species, or let's say a new bacteria, because we are interested in a bacteria study. Okay, so the other thing that we can use this strain or procedure is to check for the thickness or thinness of a peptoglycan that we have. Remember in the previous video we talked about peptoglycan, the composition, everything like that, where it is found. We have talked about it. So we'll be using this strain a gram procedure to check for the thickness. So to check for the thickness or thinness of what? Of the peptoglycan available layer. Alright, so peptido, alright, is from the word peptide bond, peptidoglycan. So those who can forget, you master it like that. So a peptoglycan layer is a layer between a cell plasma and also cell wall. Alright, there will be a layer. So if it is thick, that is the gram negative. So in terms of bacterias, in terms of this peptoglycan or in terms of layer, we have two types of bacterias. So types of bacteria in terms of layers. Alright, so in terms of this layer, so there's what we call a gram positive, alright, and also a gram negative. Okay, so in terms of layers, we have two types of bacteria. Okay, we have two types of bacteria. So there's gram positive and gram negative. So the first one is the gram positive, the second one is gram negative, like that. All right, okay, so if it has a gram positive, meaning it has a thick peptoglycan. All right, so thick peptidoglycan. So if it has a negative, gram negative, meaning it has what? Thin peptidoglycan, like that. Okay, so these are the types of bacteria that we have in terms of their layers, or just the layer between the cell wall and the cell plasma. Okay, so there are things needed in the gram procedure. Okay, there are things needed in this gram procedure. So say things needed. Okay, so the things needed, let me just go back. So these are the things, you can just write them down. Okay, so there are things needed in this study. All right, we can't just be, so there are things or reagent which are needed. So we'll talk about reagents now. All right, so the first thing that we'll talk about is, please know them in this order that I'm listing. Okay, so there's what we call a primary dye. Now this is these are things needed. Okay, primary dye. Okay, so this primary dye will be using what? Crystal violet, like that. So the second reagent, you'll be using them the way I'm listing them. So the second reagent is called a mordant. This will be using what? Iodine. All right, we all know what iodine is, and crystal violet. So then the other reagent that we'll be using is what we call decolorizer. Okay, So this will be using what we call alcohol. All right. So the other things now, there are just four of them. There's what we call now crystal violet, which will be just using what um counter strain. So say, let's say counter strain. Okay, it's a stage counter strain, which will be using safranin. Okay, so it will be using safranin. Okay, so these are the things, so know them in this order, please. Know them in this order, please. I beg. Okay, so now these are the things needed. So how do we use them now? So the first stage, so we'll say stages now. All right. So what you do is there will be a food sample that contain that, a food sample is always there. So in short, not really food sample, but we say a sample, okay? Because we are getting a sample from, let's say, a certain soil wound. So what we are going to do is we are going to flood that sample with the crystal violet. So we start with crystal violet. All right. So flood, you say flood. So say flood the sample so we're going to flood the sample with a crystal violet. Okay, so with a crystal violet, okay, for just 60 seconds. All right, so you flood this sample, the sample that you have, with what we call a crystal violet. So the results now will be, so now these are the results. Okay, so the results will be all bacterias will turn purple. So all bacterias purple. So all bacterias are purple. So purple dye. So meaning, what we call this is now purple dye. That's why we call it primary dye. So this is what we call now, it will be positive. All right. So in terms of colors, so let me make it clear here. Once they turn purple like that, okay, simply means it is too thick. Okay, so it means it is thick. So purple means positive. All right, but colorless, pinky, and red, these are what? Negative. Okay, okay, so that's stage one. So all bacteria are purple dye, meaning positive. All right, so that is the stage number one. What about stage number two? Stage number two, you say, okay, so stage number two, you say, add the way they were moving here, the way they, so we now go on iodine. So you master the reagent like that, so that you know how to do it. So once you flood the sample with the food, they will all turn purple, right? That's positive, meaning there is still a positive, meaning they have thick glycopeptoglycan. So now once we are done with stage number one, you go on this stage or step number two, you add iodine solution. So the first one was using crystal violet, the second one is the iodine solution. So add iodine So you just add the iodine for how many seconds? You do the same 60 seconds for stage 1 and stage 2. So this iodine, it uses what? It uses what? Mordant. So that's what we call a mordant stage. Alright. So a mordant stage. There will be a deoxyrize stage, mordant stage. So the results will be now, what would be the result? How would we get the result now under this? So the results will be now after adding iodine solution, all purple. Alright. All purple. You are done. You go on stage. So once they are all purple, you know what it means. I can't explain that. You know what it means by now. So this is now stage 3. So stage 3, you add alcohol. This alcohol, a stage is called the decolorizer or just the decolorizer. So the alcohol will be used to decolorize. Alright. Then the iodine solution will be used to mordant that sample we have. So meaning so that it can adhere to the wall. I mean to the cell. So forget about that. Just know the way what I've written, and also we are adding now what? We are adding alcohol. Okay, this alcohol is called the decolorizer. Okay, we were there to remove the colors that were all purple. Okay. So we'll do this for about 20 seconds. Alright. So what will be the results now? We go to the results. Once we do this, what will be the results? Okay. So the results will be, for those with the gram positive, they will turn purple. Alright. For those with gram negative, they will turn what? Colorless. That's under stage. Please just master these. Alright. So they will turn colorless like that, meaning they are gram negative. Gram positive, they will be purple. Okay. So the last stage, which is stage 4 now, you say so for stage 4 you add saffron in, which will be counter stain. Okay, so we're add saffron in. Alright, so this saffron in is called counter stain. Okay, the stage is called counter stain. You counter stain by adding saffron in. Okay, this should be performed between 1 minute to 2 minutes, so that you can have the best results. So under this again, I just want to conclude, show your results now. So gram positive, it will be now again purple. You get the point? Then gram negative, it will be what? Pink or red, like I described in the other part. Pink to red, like that. So meaning that they are gram negative. Okay, so that is it. Please, this is the end of the lesson. Make sure that you master the stages, procedure, and also the reagent in it. They say, can you write a gram procedure or gram stain procedure? So you'll be able to write all these, and you'll be given something like 6 marks or 5 marks, so if you were to say. So master the stages. The first one, you flood the sample with the crystal violet. All bacteria will be purple. In that case, add iodine so that they can adhere to the cell wall. So you add iodine solution for at least 60 seconds. That's what we call a mordant. A mordant is more like you are touching. You want them to get thick. Then for the stage 4, stage 3, you add alcohol to decolorize, remove all the colors, and see how also it will perform in terms of thick or thin peptoglycan. That will give you what? A purple or a colorless for negative. Then add saffron in for to just counter stain. You conclude. So a gram positive will be purple again, a gram negative will be pink or red. So this is mark the end of the lesson. Please subscribe to the to this channel so that you can be updated whenever I post a video. It's a stage by stage, so when you miss, it's up to you. So it's better you subscribe to these videos and also be updated on the new things we are posting. Thank you very much for watching the video. Please subscribe.